Okay, welcome. We're here on the...
Hi, I want to welcome everybody to this very special webcast. I'm Mary Miller from the Exploratorium.
And I'm Ron Hipschman.
And we're very excited to have a class here at the Exploratorium from UC Berkeley, an oceanography class. We're going to be connecting with a ship that is 4,000 miles away from San Francisco on the island of Grenada. Today we're going to be talking with a geologist, Steve Carey, and a biologist, Lee Marsh, who are both on this very special ship called the Nautilus. Let's see if we can get them up now. Steve and Lee, do we have you? We had you a second ago.
Hi, Mary.
Hi.
Welcome. We're here on the fantail of the Nautilus, here in the Lesser Antilles volcanic arc. And we're situated right over the crater of Kickem Jenny submarine volcano. It's one of the most active submarine volcanoes in the Lesser Antilles. It's got a very unusual name, Kickem Jenny. It refers to very strong currents that are present directly above the volcano, and the fishermen who used to fish in these waters likened it to being kicked by a jenny. A jenny is the word for a mule here in the West Indies. So we've been having a very exciting expedition so far. We're very happy to join you. This is Lee, to my right.
Hi there. My name's Lee Marsh. I'm a biologist on board the Nautilus for this leg.
So we've been exploring Kickem Jenny submarine volcano. We've been collecting gas samples and taking temperature measurements and doing some new work with an instrument called the laser spectrometer, which we can use to determine carbon dioxide and methane concentrations coming out of the volcano. One of the objectives of the work is to see whether the volcano might become active again. This volcano erupts about every 10 years, and the last time it erupted was 2001, so we're overdue for an eruption. And we think something might happen fairly soon, not too soon, because we have Nautilus parked right over the volcano right now. But many of the scientific objectives that we're going to try to accomplish have to do with a sort of monitoring of the volcano. What is the state of the volcano, what it's likely to do in the future?
Well, thank you, Steve and Lee, for that update. Steve, I wanted to ask you, because the Nautilus, the ship that you're on right now, is a bit different than a normal oceanographic research vessel, and your mission is not quite maybe the same as the students here might envision that they'll be doing when they become full-fledged scientists that go out on ships. Can you describe a little bit to us what the Nautilus is capable of and what makes it different than, say, a ship you'd use for an NSF or a NOAA research expedition?
Yeah, the Nautilus is very different because it's equipped with what we call telepresence technology. So a lot of the operations involve remotely operated vehicles or robot vehicles that are deployed on the seafloor. And in order to make observations on the seafloor, we have video cameras on board the vehicles. And that video signal comes up to the surface via a fiber optic cable, and then is transmitted by a big satellite dish behind me over here up to a satellite, and then beamed down to the Inner Space Center at the University of Rhode Island to be distributed over the internet too. So essentially what happens is this cruise allows almost anyone to tune in and join the excitement of discovery. This is different from normal oceanographic ships. Most ships go out with a limited number of scientists on board, and they go out for long periods of time. And so when you make a discovery out at sea, oftentimes many people don't get to appreciate it until days or months later. But now with telepresence technology, we're able to bring the excitement of discovery right into everyone's living room, basically, and we increase the scientific staff by millions. We basically have a worldwide audience viewing what we're doing. So it's very different from a traditional oceanographic expedition. The other difference is a fundamental focus of the Nautilus work is educational outreach. So we're really trying to engage the public and students in the excitement of oceanographic research, the importance of oceanographic research, and we can do that very effectively using telepresence technology.
So Steve, you mentioned the capabilities and a little bit about the ROVs. We have some footage here that actually shows the Hercules as it does its work. Can you describe a little bit besides just exploring the ocean bottom with cameras, what is the Hercules capable of?
Well, Hercules is an absolutely remarkable vehicle, weighs about 5,000 pounds. It operates in tandem with another vehicle called Argus, which is right here. So a two-vehicle system. Argus is connected by the fiber optic cable to the surface ship, and Hercules basically is connected to Argus by a tether. The advantage of that is that Argus takes the motion of the ship, and Hercules can remain almost stationless. Hercules has a variety of tools on it which we can use to explore the ocean floor. One of the most exciting tools for me as a geologist is that it has manipulator arms. So it can actually grab samples. I like to pick up a lot of rocks when I'm down there. It can also take push cores in the sediment. This cruise, we've adapted it to have a number of instruments, geochemical instruments that are measuring the composition of the water and the gases that are coming out of Kickem Jenny. It's also equipped with a and that's kind of the fundamental importance of Hercules, is it allows us to see with great clarity what the seafloor looks like. So a typical dive will involve launching both vehicles, going down to the bottom. We have some scientific objectives to accomplish. And we have two pilots. One is operating Hercules, and one pilot is operating Argus. It's very much like a very well-orchestrated dance. They work very closely together as they move the vehicles over the seafloor.
So Steve, you are an expert on volcanoes, and I know you study both volcanoes on land and volcanoes underwater. I want you to describe for us a little bit about the difference between those kind of volcanoes. I know some of them can be quite explosive. We have some footage of West Mata, and it looks pretty dramatic. But oftentimes, like with the volcano you're on now, it's a little bit quieter. So can you tell us a bit about the difference between submarine volcanoes and volcanoes on land?
Yeah, well, the prime difference between volcanoes on land and volcanoes in the water is the environment in which the magma from the volcano is injected into. So when volcanoes erupt on land, magma is ejected either into the atmosphere on land, and it cools usually much more slowly than magma that's erupted in the ocean. So when magma is erupted in the deep sea, it tends to quench extremely quickly. And what that means is lava flows on the ocean floor are unable to travel large distances. On land, lava flows can travel several to tens of kilometers away from a volcano. But under the sea, they essentially pile up very close to the vent because they're cooled off extremely quickly. It's certainly possible to have explosive eruptions underwater, and I think you're showing a video of the West Mata eruption. But even with explosive eruptions underwater, the great pressure of the water column over the volcano essentially acts like a dampening mechanism. So the intensity of the eruptions is greatly reduced underwater. One fact about underwater volcanoes that I always like to tell people is that, and they don't realize, is that 3/4 of the volcanic activity on Earth occurs underwater, not on land. So I think most people view volcanism as something that occurs on land, when in fact most of it's occurring under the ocean. And that volcanic activity actually builds the ocean basins. So volcanism is a fundamental part of development of the oceans throughout the world.
So Lee, I had a question for you. You're a biologist, but you've specialized in really trying to understand how organisms adapt to deep sea environments. They can be very harsh. There's underwater explosions and hot gases and chemicals. How does life survive there? What are you studying that makes this life kind of special?
The first thing to talk to you about is the deep ocean, really, and what the animal life in the generic deep ocean is. It's very deep, it's very dark, it's very cold, so nothing can really survive. There's no phytoplankton, there's no sunlight, so there's no photosynthesis. At these areas of hydrothermal vents and cold seeps, we have animals which survive off of this process called chemosynthesis. So whereas photosynthesis uses the sunlight to create organic compounds and carbohydrates, these animals use bacteria, and that bacteria uses chemical energy, therefore chemosynthesis, to make these carbohydrates within the... So that's the bottom of the food system at these hydrothermal vents. And as you say, these areas are very toxic, they're very hot. The animals don't actually live in the super hot waters, but they live kind of just around it. That temperature dissipates really quickly within a matter of centimeters. So when you see us getting temperature readings around 100 degrees Celsius, the animals are actually only living in sort of 20 to 30 degrees, so like more of a warm bath, really. And what they do is they either eat directly the bacterial mat. So if you've been watching us on Nautilus Live, you would have seen a lot of white material over the tops of the hydrothermal vents at the volcanoes. So some of those animals would have been directly grazing off of that bacteria, much like how, I suppose, a cow would eat grass in a field. And other animals actually harbor their own chemosynthetic bacteria. So some of the shrimp, for example, they will actually have bacteria within their gill system. And what they do is they'll waft these special modified gill filaments, and they'll actually consume the bacteria from there. So, I mean, it's very different life, and that's what creates these nice little oases in the deep ocean. I mean, we've been very fortunate to find the hydrothermal vents on top of the volcano at Kickem Jenny, but also the cold seep environments down on the avalanche slide, down at 2,000 meters, where we found lots of large mussels up to about 35 centimeters, nice big clam bed areas, and all sorts of life, really. As a biologist, it's really spectacular to see.
So we've been showing some footage. We're showing some footage now from Kickem Jenny, showing some of the bubbles. I guess you're on a area right now that has a lot of bubbles and bacterial mass. So tell us just a little bit more about life around this volcano and what you're doing on this cruise right now and over the next week or so.
Okay, well, what you're seeing at the top of this volcano, we have seen a number of chemosynthetic animals. There's a few areas of like shrimp which are specifically chemosynthetic. But I think, again, for me as a biologist, the really interesting thing is to see the number of large fish and loads of krill within the water column. Because this area is so productive, because of the volcano and because of all of the bacteria that's there and all of the krill that are living there, we're actually attracting quite a lot of large predators. So we've seen quite a number of almaco-jack, a few tuna, some grouper and things like that. So we're actually seeing an increased abundance of what we would call normal pelagic biology within this area.
One of the very interesting things about Kickem Jenny is that the gas that's being given off at this volcano is carbon dioxide, probably about 96% carbon dioxide. So one of the effects of discharging carbon dioxide into the ocean in the crater of Kickem Jenny is that it acidifies the water. Normal seawater has a pH of about 7 or 8, but we've taken some measurements here at Kickem Jenny, and the pH is as low as 4. So the waters are highly acidic. And what this provides is a little natural experiment in kind of the extremes of ocean acidification. We can see the effects on the biological organisms, albeit these kinds of pH changes are much larger than what we expect in the big ocean. It's very interesting to be able to study how the biological communities do react to acidic waters. And this is a great place to study that. So it's a great kind of symbiosis between geology and biology in the same place.
Great. So I was on the Nautilus a couple of weeks ago, and one of the exciting events there, besides when the ROVs get launched in the water, is when the ROVs come back, because they bring samples with them from the geology and biology. Can you tell us a little bit about that process? And we do have some footage we can show of the vehicle coming back on deck and collection and processing. So take us through that.
Okay, absolutely.
This is Lee's job.
So as you say, it is a really exciting time for us. We've been watching for a number of hours. We could be in the water for 12, 24, 36 hours, collecting all our samples, whether they be rocks or animals or any kind of biology. So when it comes on board, it's the first time we actually get to see these animals. So for us, it's kind of like Christmas comes at once. We all rush to the vehicle. We have to wait until we get the go-ahead. And then the most important thing for us is to make sure we know exactly which animal has come from which compartment on our vehicle. It's very important that we can tie each animal, each rock sample to the environmental data. So we need to know what depth it's come from, we need to know the position, we need to know the oxygen concentration, the temperature, all these kind of environmental factors. So we can tie that back to the environmental situation that those samples have been collected. So once the vehicle's on deck, we will all hover around, we'll get our little buckets, and they'll all be labeled up so we know exactly which sample is which sample within the tool tray itself, and then we'll move them into the wet lab. Now we have a wet lab on board. It's quite small, but it's perfectly formed. It means we can actually process all our samples. So in Steve's case, we'll get the rocks, and they'll be smashed into subsamples so they can go off to different institutes to look at different chemical compositions. And for me, with the biology, what we'll end up doing is once the specimens come on board, we'll take a tissue sample. Now that first tissue sample, that will go for DNA analysis. That means we can work out what species this is. If we have a number of them, we can start looking at population genetics. So we can start looking at how related this species is to similar species at other hydrothermal vents or cold seep environments. The rest of the specimen can get preserved in a number of ways, depending on what the scientists ashore would like. So we might have scientists that are looking at reproductive ecology, so they might want to take specific bits of tissue for that, or what the specimens are eating, so we could freeze some of the tissues for that. And also we send a whole specimen back to the Museum of Comparative Zoology at Harvard, so that we have a sample for taxonomy and a voucher specimen essentially from this area. But generally it's a really exciting time. It's one of my favorite times anyway.
I guess that's another important aspect to highlight about the program is unlike on some other oceanographic cruises, all the samples that we collect here on Nautilus and all the data are freely available and open to the public. So this is a kind of hallmark of this program. It's a completely open exploration experience.
Great. Well, thank you so much. I want to remind the audience online that we're talking live with EV Nautilus. It's a ship operated by the Ocean Exploration Trust, currently diving off the coast of Grenada. We're talking with a biologist and geologist on board, and we want to welcome any of the questions from the online audience. You can tweet them to #EVNautilus. You can also go to the Nautilus Live website when they're diving. They take questions from anybody online, and they'll answer them as they're diving from the control room. I want to see if there's any audience questions here at the Exploratorium for Steve or Lee. We have a question right here.
Yes, I was wondering how, when you're setting up and gearing up to go out on these dives and these explorations, how you choose the areas in which you're going to explore.
A lot of the way we choose the dive sites, Nautilus is equipped with what's called a multi-beam sonar system. And this system allows us to map the ocean floor with very high precision, so we can develop three-dimensional maps of what the ocean floor looked like. And so commonly, one of the first operations that goes on is to develop a map for the area, unless we already have one. So we do the mapping on board the ship. We generate a nice three-dimensional map, and then we use that map to select dive targets based on looking at the topography of the bottom.
Steve, I have a question about what it's like for you as a scientist who goes out on ships but also has participated from shore. Do you feel like there's something that might be missing if you always were on shore as opposed to being out at sea? Tell us a little bit about that. I think people in the audience here from UC Berkeley might be interested in what kind of future work they might have.
Question. I think for me personally, I think a combination of both is kind of an ideal world. I certainly enjoy going to sea. I mean, I absolutely love it out here. You develop a really nice camaraderie with the people that are aboard the ship. You meet a lot of new friends. It's extremely hard work. We get very little sleep during a cruise. But there are often times when other responsibilities, like for me personally, teaching or other responsibilities preclude me from going on a cruise. But with telepresence, then I'm able to participate remotely. So it basically opens up the number of opportunities in which I can participate in oceanographic research by combining both going to sea and using telepresence as a virtual cruise, essentially.
Do we have some more questions here from the audience?
I've been joining in from ashore.
Lee, go ahead, please.
Sorry, no, that's fine. I was just going to say, for me as a young scientist, I've joined in from ashore, and it's been a fantastic opportunity for me to work with other scientists that I might not necessarily get to work with or communicate with. So having that telepresence and being able to be involved from ashore as well has been fantastic for me personally as well. It's been great.
Great, thank you. Are there any other questions from the audience here? We have one in back.
How did each of y'all get involved in y'all's respective fields, or what drew you to them, or what you're interested in? Or, yeah.
So my interest in volcanology evolved from, I got an undergraduate degree in geology. I've always been kind of interested in the outdoors. And when I went to graduate school, Mount St. Helens erupted in 1980. Of course, I'm dating myself now, but I had an opportunity to go out to Mount St. Helens and about 1 week after the eruption to see the effects of this event. And that was my sort of aha moment, seeing the tremendous power that nature holds in volcanoes, and I was hooked. And from then on, volcanoes are essentially the only thing that I've wanted to study.
And for me, I actually did an undergraduate in oceanography and marine biology at the University of Southampton in the UK. And I was giving a very inspirational lecture by Professor Paul Tyler, who's a world-renowned deep-sea biologist. And I think when I had that first lecture on hydrothermal vents, I was convinced this is all I wanted to look at. But unfortunately, trying to get a PhD in hydrothermal vents wasn't the easiest thing in the world. It's quite expensive to come to see and to study them. And the UK itself didn't have very many programs going to sea at the time. So it took me a few years. I waited and I worked and until such time that there was a title, there was a PhD I was interested in, and where it's quite different in the UK to the US system. But I waited until there was a research cruise that was available that I applied for a PhD then to actually study hydrothermal vents in the Antarctic. And now I've been to ones in the Caribbean, the Southwest Indian Ridge. I've been very fortunate. It's been a fantastic career so far. Let's hope it's just the beginning.
Thank you. Are there any more questions here? We have a couple of questions here.
Hi, what are some unexpected things that you found around this Kickem Jenny area?
Sorry, could you repeat the question? I didn't quite hear it.
The question is, what are some unexpected findings from the area that you're diving now, from Kickem Jenny?
Oh, well, so last year to start out with, we made this unexpected discovery, as Lee mentioned, of these cold seeps that are on the flanks of the volcano. It's very typical for volcanoes to have hot fluids and hydrothermal vents because you have a magmatic system that's very hot and you circulate seawater through it and you generate hydrothermal vents. But it's less common, and it's in fact extremely rare to find cold seeps on the slope of a submarine volcano. And this is just an example of, you kind of never know what you're going to find on the bottom of the ocean. We weren't looking for them. We stumbled across them quite by accident. And it was a remarkable discovery. These are cold seeps that are formed at the end of this avalanche that flowed down the side of the volcano. And we think it essentially squeezed the fluids out of the sediment to form these cold seeps. And we find these tremendously interesting chemosynthetic biological communities that are developed on here. So Kickem Jenny is one of the few places in the world where I think you find both cold seeps and hot hydrothermal vents in the same location.
In such close proximity, yeah. I think this is one of the very first. Yeah.
So that's very exciting.
Great. Well, we have one more question here. I think we have time for one more question. Yep.
Hi. Very similar question, but for Lee, one of the really exciting things about deep sea dives is that you're actually always finding really new biological specimens. So has there been anything exciting that you have found?
Sorry, can you repeat the last half of that question? I just missed the last half.
Yeah, it's all good. Has there been anything exciting that you found in the way of biological specimens?
Okay, actually last week we did our first Hercules dive on the cold seeps. So it's the first time this cruise we were able to sample some of the clams. I don't think they'd been sampled from last year. And actually when we opened up the clams themselves, when I was getting the tissue sample out of the clams, it's not unusual to find the odd polychaete, so the odd worm inside the clam. But within every clam there was at least 15 to 20 different types of, same species, but 15 to 20 polychaetes associated with each of the clams. So for me, that was really interesting. The clams look healthy. They're obviously, I don't know if they're parasitic or what was happening, but they look very comfortable living within the gill structure of the clams themselves. So our sample inventory went up rather rapidly. When we thought we'd only collected 10 clams, it turns we collected 10 clams and maybe 120 polychaetes. So it meant it was a long night processing, but very rewarding at the same time. So it was great.
Thank you both so much, Steve and Lee, and the crew of the Nautilus, and also the Inner Space Center for taking time today to talk with us. We really appreciate it. Bye-bye for now.
Oh, so long.
Bye.
Nice to be
Thank
with you.
you.
I want to thank the audience that's here today, the Ocean Exploration Trust, Oak Meadow Foundation, and McBean Family Foundation for their support.